Now we are going to discuss about the remaining part of our first unit, that is, what are the newer trends in systematics. We know that systematics is nothing but it's a convenient system of classifying, identifying organisms for inferring their ancestry as well as evolution. Actually, the newer trend in systematics means it involves a shift from the morphological consideration to embryological, cytological, ecological, ethological as well as genetic consideration, which means at the very ancient times, organisms are classified on the basis of mostly their morphological or observable features. But now, in the case of this newer trends, we are shifting this morphological or observable consideration into much more advanced forms, that is based upon the embryological aspects, cytological aspects, ecological aspects, ethological or behavioral aspects as well as genetic consideration. We are classifying organisms. So this is called a newer trends in systematics. And this newer trends in systematics involves the following forms, that is chemotaxonomy, serotaxonomy, numerical taxonomy, cytotaxonomy, cladistics as well as molecular systematics. So as you are just recognize or just analyzing the different forms, you can easily identify what may be the meaning of chemotaxonomy. We are classifying or we are taxonomically identifying organisms on the basis of the similarities of the chemical substances. Serotaxonomy, similarities in the serum or antibodies or antigens, etc. The numerical means some number of similarities as well as dissimilarities. Based upon this number of similarities as well as dissimilarities, we are classifying organisms. Then cytotaxonomy means we are classifying organisms on the basis of chromosomal or cell features. Then cladistics and molecular systematics are another branch in which here also we are classifying organism on the basis of their molecular concerns as well as another taxonomical identities. So these are the different aspects of this newer trends in systematics. Then we are starting with the chemotaxonomy. This is a method of classifying. I told you that this is a method of classifying organisms on the basis of the similarities in the structure of chemical compounds which is present in the organisms that being classified. That means here we are classifying organisms on the basis of certain kinds of chemical substances which is present in the body. Proteins, we know that the proteins are generally controlled by genes, right? By means of the transcription as well as the translation process from the genes or from the DNA, it gets transcribed into messenger RNA, and from the messenger RNA it gets translated into the proteins. So these proteins that is mostly controlled by the genes are generally used for this kinds of comparative studies in chemotaxonomy. Apart from these proteins, we are also utilizing nucleic acids, peptides for this classification. Here the chemical features of the plants are widely used in developing classification of solving the taxonomic problems. This is the case of plants. The chemical features of certain plants are widely used for solving different kinds of species in the case of animals, in the case of kingdom planting. Then this is used for classifying organisms from a very simple to much complex forms like fungi, bacteria, and up to a highly advanced angiosperms. Then what are the different chemical characters which are generally employed for chemotaxonomy? That includes the following. First one is the direct visible characters. For example, starch, stain, silica and gypsum. These are the substances which can directly sensed by our sense organs. So by means of this visible characters we can classify organism. Then character which is known by chemical test. For example, phenolics, oils, fats and waxes which is generally produced by most of the plants can be tested or can be identified by means of chemical test. So by using this also we can classify plants. Then proteins. I told you that proteins are which are generally controlled by the genes are also used for this taxonomical purposes, and they generally includes these proteins can be categorized into two different forms: low molecular weight compounds as well as high molecular weight compounds. The low molecular weight compounds include amino acids, alkaloids, fatty acids as well as terpenoids. I hope you understood what is meant by each one of this. Then high molecular weight compounds includes most of the macromolecular structures like DNA, RNA, polysaccharides or sugars as well as proteins. So by considering both this low molecular weight proteins as well as high molecular weight proteins, we can classify organisms. Then what are the aspects of chemotaxonomy? We can develop a taxonomic characters which may improve the existing system of plant classification, and also we can develop present-day knowledge of the phylogeny or the evolutionary relationship of plants. The first one is we can develop the taxonomic character. We can develop a new taxonomic character by means of this chemotaxonomy, which will which will alter or which will generally replaces the existing system of plant classification. Then also it helps to develop present day. We can, by means of this extraction studies and comparative studies, we can increase our knowledge of phylogeny or evolutionary history or evolutionary relationship among plants on the basis of this chemical substance. So these are the two different aspects of chemotaxonomy. Then what about the significance of chemotaxonomy? We know that plants stores a wide variety of chemical substances, and these substances each have a significant role in taxonomic studies. They contain phytochemical constituents or the chemical substances which is generally present in plants, which are very much important for the taxonomical studies, and we can group them into the following constituents, that is primary constituents as well as secondary constituents. That means based upon the nature or the complexity, we can divide all these chemical substances or constituents into two different forms. That is called a primary constituents. It generally includes macromolecules that are directly involved in metabolism. For example, proteins, nucleic acids, chlorophylls and sugars. These are the macromolecules, or they are very large in size, which are directly involved in the metabolism. And secondary constituents includes those compounds which are not directly involved in metabolism, and these compounds generally lacking the nitrogen. Okay, for example, phenols, caffeines, benzoic acid, nicotinic acid and polyphenolic compounds. So these are all the significance of this chemotaxonomy. So I hope you understood. If you have any doubt, please do in the comment section. Thank you.